The broadcast is now starting. All attendees are in listen-only mode.
Hi, good afternoon, everyone, or good morning. This is George Orling. I'm the Director of Medical and Scientific Affairs at HGSA, and I want to welcome you for all to dialing into this month's research webinar. Really excited to have two presenters, Dr. Vicki Wheelock and Dr. Jan Nolta, all the way from California, presenting to us on an update from their UC Davis stem cell research program, as well as their progress on their pre-cell observational trial. Before we get started, I just wanted to remind everyone, if this is your first webinar, that we're recording this webinar, so if you know friends that would like to see it and couldn't join us at this time, within a week this will be on our website under the research webinar series. If you have a question at any time, you just go into the red box where it says questions and enter it here and send it to me, and at the end of Dr. Nolta's and Dr. Wheelock's presentation, we'll be reading them to them for them to answer. As I said, I already covered this, but we are recording this, and we'll be posting this to our national website in the research webinar section. Along with this recording, you can see all of our other archived research webinars, just in case you're interested. Along with the recording, we'll also be posting some information to our website under the clinical trials section. This is a survey that UC Davis requesting feedback from an HD novel treatment survey, and we'll be putting this in the clinical trials section of the HGSA website. This is going to help them, very similar to what HGSA has presented or had a survey out a few weeks ago looking for feedback that we want to provide to the FDA for our upcoming patient-focused drug discovery meetings with the FDA in 2015. The UC Davis is requesting feedback from the community about some of the concerns and attitudes you may have in regards to future HD treatment. So take a look at this survey, and we'll be sending out an e-blast about this as well. So finally, I just would like to introduce our speakers. We're really pleased to be welcoming both Dr. Wheelock, who is joining us as she's a health science clinical professor of neurology at UC Davis, where she's been a clinical professor since 2009, and she's also been the director of our HGSA Center of Excellence at UC Davis since 2001, and is an active member of the Huntington Study Group and the HD research community. Joining her is Dr. Jan Nolta, who's the director of the stem cell program at UC Davis School of Medicine. Dr. Nolta also directs the new Institute for Regenerative Cures at UC Davis. Dr. Nolta's current research, which we'll be hearing about, is focused on developing therapies that will use mesenchymal stem cells to deliver different factors, or in particular brain-derived neurotrophic factor, for treating HD. Dr. Nolta is also funded as the co-PI on several major grants from CIRM, which is the California Institute for Regenerative Medicine, which has the goal of developing and delivering some novel phase 1 clinical trials for HD patients. So without further ado, I'm going to turn it over to Drs. Nolta and Wheelock. Okay.
Very good. Thank you, George. And I want to thank you so much, and we both want to thank HGSA for inviting us to be part of the research webinar series. It's our pleasure to be able to talk to the community and engage with the community, and we're interested in hearing questions, as you mentioned earlier. So today's talk will be an update on our stem cell research program. We'll show you some basic science development, preclinical development progress, but we'll also talk about the observational study that's been enrolling for the last year called pre-cell. So the outline of our talk is to mention the challenge of finding meaningful treatments for Huntington's disease and juvenile Huntington's disease, to talk about the platform of using these remarkable adult stem cells as a potential therapy for Huntington's, update about the preclinical studies, and then also to give you an update about pre-cell and about our future planned clinical trial. So as many of you know, Huntington's disease is a difficult, terrible challenge to patients and to family members. It's a slowly progressive hereditary disease that has very prominent neuropsychiatric features. And we estimate about 30,000 people in the United States have Huntington's disease, but with about 150,000 additional people at risk. And we estimate about 2,000 new cases per year. There are some figures that suggest that the cost of care for patients with Huntington's disease and the economic burden to their families is $2.5 billion per year. And the symptoms are movement symptoms, cognitive symptoms, and psychiatric and behavior disorders. So this is an image showing brain degeneration that happens with Huntington's disease. On the left-hand side of the screen, you see a brain section from somebody not affected by Huntington's. And the right side, you can see somebody, the brain section from somebody who has died after having Huntington's for a long time. And listed there are a number of the behavioral and motor symptoms that we see with Huntington's. Again, it's multiple different issues that people have, striking behavior, psychiatric, and motor symptoms that lead to progressive disability. The area that is of greatest interest to us and to many researchers is the striatum. That's the part of the brain that helps to control thinking and movement and behavior. That's the area that's selectively targeted in Huntington's disease, where the mutation causes cells there to die. Huntington's passes from generation to generation, and the gene for Huntington's was found 21 years ago, a CAG repeat expansion in the Huntington's gene. And unfortunately, we still don't have any disease-modifying treatments for Huntington's, as hard as everyone has been trying. So we do have treatments for Huntington's. We certainly have the support of HDSA, support groups, counseling, benefits programs that are government and both national and state-based. We have the important advocacy of patients, families, and HDSA. We do have symptomatic treatments, and we have excellent palliative treatments to help manage the symptoms, but we can't yet slow down the course of Huntington's. And it's very exciting to know that organizations like the Huntington Study Group, the EuroHD Network, and many, many different organizations are heavily involved and invested in finding treatments for Huntington's disease. And yet, we still don't have any effective treatments that would delay the onset or slow progression for Huntington. So 2 ways of thinking about developing a treatment. One would be a neuroprotective treatment. That would be a therapy that would either delay the onset or slow the progression, maybe keep people on the earlier stages longer. If we could do that, that would be great. Even better, though, would be a cure, where we'd have a therapy that would prevent the mutation from killing brain cells. And there are a number of ways that have been proposed to do this, and we will be talking a little bit more about these gene-modifying platforms as we get further. Dr. Nolta will talk a little bit about that. So I'm going to hand it over to Dr. Nolta.
Great, thank you so much. So I wanted to start out with just going over very briefly the types of stem cells that we researchers use. So I want to first talk about, not use that mouse, okay. I first want to talk about the pluripotent cells that are on the right side of your screen. You can see those little colonies of cells, and these are the embryonic stem cells that we use to generate new neurons. And then even more exciting, as many of you know in the Huntington's community, are the induced pluripotent stem cells. And many research groups are using these. They're made from the skin cells of a patient. They start with adult stem cells, and they can be turned back in time to be much like the embryonic stem cells, and we can make neurons out of them and make striatal neurons. And so there are many wonderful groups, not ours, but wonderful groups that are working on developing therapies for replacement neurons. And I just wanted to give them a little shout out, because that's really amazing technology. What we do in our group is that we're using the adult stem cells, shown on the other side. The round cells are the blood-forming or hematopoietic stem cells, and they just make blood. But the broad flat cells on the bottom of that picture are the mesenchymal stem cells, or supporting cells, and their job is to nurture other cells. And so I've spent my career since, oh boy, I don't want to say how long, about 30 years, studying these cells, these mesenchymal stem cells, and what they can do and how they can deliver factors for keeping other cells and tissues alive, and that's what we'll really focus on today. So these are adult stem cells from the bone marrow. The MSCs repair damaged tissues by responding to the scene of the injury, the tissue injury, and they produce healing factors. And I'll show you what they do in action with some little videos. We do call them the paramedics of the body, and they have a strong safety profile in clinical trials that have been done for other diseases. Here's a video of cell-to-cell communication, as we call it. And these are the mesenchymal stem cells communicating with each other. It's kind of like their Facebook and Twitter. And we can see that they make these, hopefully the mouse works this time. Yeah, they make these projections here that are called tunneling nanotubules. And you can see little things going through those nanotubules. It's kind of like a gas hose that they put into another cell. And there are little, even mitochondria that go through. Those are like the cell's batteries, go through into damaged cells. And this is how they keep damaged cells and neurons alive. They also do another cool thing, which is to make little batches of these round things, exosomes. They leave them behind, and then other cells gobble them up, and then the other cells get energy and divide, as you just saw. And so the mesenchymal stem cells, their job in the body is to crawl around through the tissues and help the other cells that are in distress by giving them factors. And these are directly from cell to cell through the tunneling nanotubules and exosomes, very scientific words, but really they're helper cells, and they're there to help the other cells and tissues. So I realized early in my career that these MSCs could be genetically engineered to secrete really copious amounts of factors for delivery to other cells and tissues in the body, because it's what they do naturally. And if we add an extra gene, such as a gene for a RNA interference molecule for Huntington's or for the brain-derived neurotrophic factor that we're going to talk about in a moment, they would make really large amounts of it. And so my lab has worked on that since 1987 on teaching these cells to make other factors, and we published a book on it in 2006. And a paper about using these cells for Huntington's disease in 2012. And I wanted to point out that this is with our wonderful colleague, Dr. Gary Dunbar, and his lab, that we've been doing this work. So brain-derived neurotrophic factor. This is a lead candidate for HD treatment. And I'm going to, it causes the survival and function of striatal neurons that's dependent on brain-derived neurotrophic factors. The mutant Huntington protein blocks production of BDNF at the RNA level, and levels of this trophic factor are significantly reduced in the brains of HD patients. In Gary Dunbar's lab, Day et al. Showed that mesenchymal stem cells engineered to overexpress this BDNF really slowed the progression of HD in a transgenic mouse model. And we know that BDNF delivery triggers the recruitment of new neurons in HD transgenic mouse models. So these MSCs are our candidate for delivery of the brain-derived neurotrophic factor. We know that these MSCs secrete neurotrophic factors, reduce inflammation, reduce programmed cell death, enhance connections between neurons, and reduce cell toxicity. As I mentioned, they can be readily engineered using viral vectors to robustly deliver growth factors and other factors, such as RNAs. The vectors do not integrate into the host cells, so the vectors won't get out of these cells into the brain. They're just teaching the cell to do new tricks. MSCs don't require immunosuppression. They have like a force shield around them where they can't be rejected. And so also, unlike embryonic or pluripotent stem cells, MSCs have a strong safety profile in clinical trials. And those embryonic and pluripotent stem cells are just getting going in the clinical trials, but MSCs have been tested in clinical trials for other diseases for about 10 years now. And there are 43 published peer-reviewed proof-of-concept studies that have demonstrated efficacy for MSC BDNF or MSC BDNF in HD mouse models. So we do this work in the UC Davis Institute for Regenerative Cures. This is a facility that's funded by the California Institute for Regenerative Medicine, or CIRM. And we're very lucky to have CIRM in the state of California, and they fund a lot of our research for novel clinical trials, and they were voted in by the voters as Proposition 71. So our facility opened in March of 2010. It's a 109,000 square foot renovated historic building funded by this third major facilities grant. And we currently have over 200 scientists, students, veterinarians, and MDs working together in this building. And this just shows the pipeline that we go through for any a development candidate that we want to take forward into clinical trials. So it starts with basic research. It then goes through translational research in the lab, preclinical research that we also call IND-enabling studies, and then we hope to start a phase 1 clinical trial that's a smaller number of patients, and we're really looking for safety of the cell and gene therapy. And then it would go to a phase 2 and even a phase 3, a very large trial. At that point, we hope that it would be approved for use in patients as standard of care. And so that's really our goal with these therapies, these novel therapies. We have the MSCs delivering inhibitory RNA against the mutant HTT allele. Further back in the pipeline, what we're mostly going to focus on today is our disease team grant that we have for MSC delivering brain-derived neurotrophic factors.
So we were fortunate to form collaboration, Dr. Knowlton and I, back in 2009, looking to develop treatments for Huntington's disease, and based on her work from previous CERM grants and other grants, using this amazing platform of using these mesenchymal stem cells, we were invited to apply for a disease team grant in 2011. We developed this project and submitted that grant to CERM, and we were very proud to be selected for a disease team grant back in 2012. The objectives of our grant are to use these mesenchymal stem cells engineered to produce BDNF as a potential treatment for Huntington's disease. First of all, to complete all of the toxicology, manufacturing, safety, all of the other studies that need to be done in order to enable us to get an investigational new drug license from the FDA to go forward into a very early phase 1 trial testing these cells in patients with early stage Huntington's disease. So the project plan for this grant was to take 4 years to go into this process. In year 1 and year 2, there's heavy preclinical work going on in the lab using transgenic mouse models, collecting all the data and putting together all the information that the FDA will need to go forward to looking at our application for the IND. However, we decided that in the first 2 years we would enroll a cohort of people in an observational study who would be potential candidates to go forward into the planned stem cell trial in the future. That study is called PreCell, and PreCell was started a year ago. We're currently following patients now every 6 months, and I'll talk a little bit more about that. And our hope is that when we get the IND in the future, that we will then move forward into our stem cell trial, and I'll be saying a few more words about that toward the end. So this is the project plan. We have a huge team of people that are working together. On the left side, you see the preclinical team, and on the right side, you see the clinical team. But we meet every week. We work together constantly. We have wonderful partners here at UC Davis, and our executive team is Dr. Knowlton, myself, Terry Temkin is our co-clinical lead, and Geraldine Annette is the project manager on the preclinical side. We have a team of people here from UC Davis. We also have consultants in other institutions that are, in our opinion, the world experts in the areas that we've asked them to consult with, and we're very proud to be working with them and going forward. So this is the idea that the mesenchymal stem cells will be harvested from a donor, a healthy donor. They will be taken over to the lab and expanded and tested. They'll be gene modified and then produced and ready to implant into patients. And the implantation, there's a little heads up there, will be through a neurosurgical procedure to implant them directly into the brain. The target will be the area where the most damage is happening in Huntington's disease.
Thanks, Vicki. So back to me. How do we manufacture these paramedic MSCs that produce factors to help damage neurons and other cells? As Dr. Wheelock had just mentioned, we harvest the bone marrow from a healthy donor who is not affected by Huntington's. We grow these MSCs up following a standard operating procedure that we have already established in our wonderful good manufacturing practice, our clean room facility here at UC Davis. We engineer them to produce extra BDNF. They already make some, but we want them to make a lot, or RNA inhibition in the future for our future planned trials. And then we freeze them until the patient needs them. And I have a little pictorial of what that all looks like. So these are the flasks of the MSCs growing in an incubator that keeps them at body temperature. It's like we have little aquariums with little cells growing in the bottom, and we take care of them daily. They divide on the bottom of those flasks under the media that has all the proteins and sugars to feed them to make more cells. And this is what they look like when we look down through the flask using a microscope. And we grow them up to larger numbers following standard operating procedures or SOPs, and then add the extra DNA to make BDNF. I wanted to point out two of our most wonderful team members in the lab who have been our study directors and are designing this laboratory work, so Carrie Pollack and Heather Stewart, and they bank the cells and test them in mice. So you can see they have a whole rack of the labeled cryo vials where the cells are going to be put into those vials and taken to the freezer. I wanted to point out how dedicated these two women are. Carrie worked in the lab to help HD and JHD patients until she was ready to deliver her baby just a couple of days before. You can see that she's very pregnant in this picture. She's wonderful. Heather just got married and only took off 2 days for the wedding and postponed her honeymoon for the cause. So a big thanks to these girls. They're amazing. This is what the cells look like. This is what we call a tox batch for testing in the lab. And the cells are in their cryo vials right before being taken quickly to the freezer. And then in the clean room facility, the environment and temperatures are monitored daily. We make sure the cells are living happily in their tanks under liquid nitrogen, frozen, until we need them. And they're then thawed out and used in experiments to support an investigational new drug application to the FDA. So the current studies that we're working on are assessing secreted BDNF levels, genetic stability, safety studies, retention studies in immune-suppressed HD mice, what are called pivotal efficacy studies, and dose-finding studies. We can show that the MSCs, after they're genetically engineered, make a lot of BDNF. We can see that as we add more of the BDNF gene to them, the amount of BDNF produced increases nicely. And then I wanted to say just a few words about our HD and JHD mouse model studies. We used a couple of strains of Huntington's disease mice. So we used the YAP128 strain of mice as a model of adult HD, and the R62-120 model, which has 120 CAG repeats, as a model for juvenile HD. The YAP mouse has late onset, and the R62 mouse has early onset and seizures. Both strains perform progressively worse at walking on the slowly rotating rotarod and other functional and behavioral tests over time. And we test to see if our therapies improve these deficits in the mice. I wanted to point out our amazing clean room vivarium team. We have a shower-in vivarium to work with these mice, and this team cares for those fragile HD and JHD mice every day, and I wanted to thank them. They're very incredibly dedicated. And, you know, it's hard to work with the mice. They're having seizures and they're not healthy little mice. And they really keep them going as long as they can. And we really, really appreciate them. So we engineer the human MSCs, and you can see them here in green, to make BDNF, inject them into the mouse brain. So they're showed here in the striatum. Excuse me. And the blue cells are the nuclei from the neurons, and they all communicate with each other in the brain. This is another picture of the green cells in the Huntington's mouse striatum. This was done by Carrie. And so each transplanted human cell will make a certain level of BDNF. This is needed, of course, to keep the neurons alive and connected and to grow new neurons in the brain. And so we hope that each one will make enough BDNF to make a nice wave of BDNF in the brain for as long as the human cells are there. The human cells do not divide and make more of themselves in the brain. They know when to stop dividing. Their job is just to sit in there and make the brain-derived neurotrophic factor or other things that we've taught them to make. So we're looking at retention and safety of the human MSC BDNF product in the brain over time, and we use the in vitro imaging system in anesthetized YAP128 mice. And so these mice are just asleep. They have some isoflurane in their nose cone to keep them asleep so that we can take a picture of them so they're not wiggling around. And we can see the cells as those bright dots that are in the left striatum of their brain. And we can monitor this over time to see how long the cells are still there and working and alive. We do striatal volume measurement by micro-CT in transgenic mice. We're looking for preservation of striatal volume by the human MSC BDNF product as our primary endpoint. And the next is a video done by Haley Nelson and the UC Davis Mouse Imaging Center team. So here is the video, and let's get to it. It takes just a minute to start going because we're cutting through the embedding matrix, and then you'll start seeing that it's going through the brain, and it will very nicely measure the size of the striatum, and we get volumetric measurements from this. So thank you to our great CMDI team and to Haley for having this as her master project. We're doing behavioral studies. This is an open field study, so it's a spontaneous movement assessment of the mice after we put the MSCs engineered to overexpress the BDNF into their brain. And this is a kind of a test to see how much movement they do and how curious they are. As the mice get more sick, they're very apathetic and they'd rather sit in one corner and not move around a lot. So we can see the blue arrow is the wild type mice. The red arrow is the red squares and lines is the transgenic mice that are sick. And you can see that after we put our MSC BDNF into the brain, the green line is almost back to wild type in this initial study. So that's what we're looking for in these different behavioral studies. We have different banks of studies, rotarod, different types of tests, and we're looking to restore the sick mice back closer to wild type. This is an analysis of brain changes in mice receiving transplantations of MSC BDNF, and showing in the green that the MSC BDNF treated mice had greater striatal optical densitometry, showing a cytochrome oxidation. And these studies have been done by Kyle Fink, Whitney Carey, and Haley. And this is on the right is Kyle Fink, who is an outstanding postdoctoral fellow in our group and has been leading a lot of these studies with the brain change. He came to us from Gary Dunbar's lab at Central University of Michigan, and we're very grateful and continue collaborating with that team. We're very grateful that Kyle came to join us. So just to summarize the efficacy studies, I wanted to show on the left here this little rotarod. That's what we asked the mice to walk on. It's just a very slowly rotating drum, and they walk on it to show their movements and how coordinated they are. And so our initial data, in conjunction with previous reports, indicate that MSC BDNF implanted directly in the brain can prevent motor symptoms and brain cell loss in mouse models of HD. And then the current studies, as I mentioned briefly earlier, we're looking at secreted BDNF levels, genetic stability, safety studies, making sure that the cells are sound and safe when we put them in the brain. Those retention studies, looking at the mapping in the mice that are asleep temporarily, these pivotal efficacy studies, and also dose-finding studies to see how many cells we should put in per number of neurons.
Yeah. Okay. So thank you, Jan, so much for going over that. So these are the studies that are going on in the lab that we will be putting together all this information to go forward to the FDA, and we have larger numbers of studies and larger numbers of animals so that we can carry this work forward. But the goal is to learn as much as we can about these cells, to engineer them, to get them ready and safe to be tested in patients. So the cells, as Dr. Nolta said, are going to be manufactured in the good manufacturing practice facility here at UC Davis, which is a state-of-the-art and I think state-of-the-planet facility that was built because of funding from CIRM to help scale us up and get ready. And there are a number of different trials that the GMP facility is already producing cells for in other studies and other diseases. So now I'm going to switch over and give you an update. You've heard about the preclinical studies and the mouse studies, and now I'm going to talk a little bit about what's going on with patients. So I already mentioned that we have 2 planned studies. One is currently in progress, and that's called PreCell. And in PreCell, we're going to enroll between 26 and up to 40 patients with early-stage Huntington's. We ask them to come in and see us periodically for very detailed neurological examinations, cognitive tests, brain scans, spinal fluid studies, so that we can establish a baseline and a rate of change for each one of these measures for every person in pre-cell. And if we are successful, and we plan to be, going forward to the FDA for the IND, the license to go forward in humans, then we plan to do a second trial, which is the HD cell. And this will be actually testing these engineered mesenchymal stem cells in patients who have early-stage Huntington's. And we expect that people who are in pre-cell, who've been very carefully coming to all these study visits and letting us to establish a baseline, will be offered the chance to go forward into HD cell if they choose to do so. We'll have a very good baseline that will help us to make sure that we can assess their safety after the cell implantation, and also give us a chance to get a sense of whether we might be also helping the symptoms of Huntington's disease. But I do want to qualify this, that this is a phase 1 trial. This is a trial at the very first step that we have to, first in man, putting these cells into the brains of patients with Huntington's disease. So the pre-cell study is a longitudinal observational study where we're enrolling this group of patients. They come in, they have vital signs, blood tests, MRI scans, cognitive tests, psychiatric assessment. It takes 2 days to complete every visit, and they come in every 6 months for these tests. This is a slide showing what the inclusion criteria are for pre-cell. So we're doing this in adults only. We have a huge challenge with JHD, but we've decided with the first-in-man trial that we would like to test this first in adults. People need to be English-speaking so we can do all the testing, the cognitive testing and behavioral testing that we need to be able to do to assess their safety. They have to be able to give informed consent and able to participate in all the study procedures. They have to have a diagnosis of Huntington's disease confirmed with genetic testing. So we're not doing this in people who have not yet been diagnosed with Huntington's disease. They have to be in the early stages of Huntington's. Reason for that is that this treatment of delivering BDNF, we know, is only going to be effective if we go early in the disease. If we wait into mid-disease or late disease, there's really very little chance of helping patients. They have to have clinically definite signs of Huntington. They have to have a caregiver or informant who can also accompany them to their visits and to give us feedback about how the person in pre-cell is doing. And this is also another measure that we're using for safety for going forward into the planned HD trial. And then we also are asking everybody who's in HD cell that they must agree to have adequate birth control measures, because we don't intend to test this treatment in a fetus, for instance, if somebody decided to get pregnant. They have to agree during the study to avoid pregnancy and to avoid fathering children for men. There's a full list of the exclusion criteria on the clinicaltrials.gov website, and I've included a link there where you can take a look at those. But in essence, the exclusion criteria are people who have unstable medical or psychiatric problems or other neurological conditions that could be potentially compromised by participation in this study. So this is a table showing what the patients are doing when they come in. So they're coming in for a screening visit, which lasts, as I said, over 2 days of time. If they meet all the safety measures that we put into the screening, they'll come back within 60 days for a baseline visit. Then they come in every 6 months, and we repeat this over and over again. We call them every 3 months in between visits so we can assess to make sure they're not having any health changes or other safety issues that might come up during the trial. So the update pre-cell was approved by the UC Davis Human Subjects Committee or Institutional Review Board. Last year, we were renewed in June of this year. We've screened 27 patients, and we have 22 enrolled to date. We are still enrolling people in pre-cell. We're doing these detailed assessments that I've mentioned for every subject who comes into the trial every 6 months. This is just a peek at some of the imaging data that we're getting from the patients who are in pre-cell. The figure on the left is what's called a template, and that's a template of patients who have normal, healthy aging, age 70 years. And then what's overlaid on that is the data from the patients who are in pre-cell, taking a look at what their brain sizes look like. And the areas that are blue or green, the cooler colors, are areas where there is substantial decreased signal coming from the brain in the patients with Huntington's compared to patients who don't have Huntington's. The areas that are red on the outside that are a little bit warmer colors or brighter colors are areas where the patients actually have larger volumes in the brain, and that's because they're younger. They're being compared to people who are older. So we can see very, very easily within the brains and the patients that are in pre-cell that they already have significant changes in the striatum and some of the connecting areas within the brain. And this will be the target of our stem cell treatment. So going forward, we're still enrolling patients in pre-cell. We're still continuing the preclinical studies, getting ready for an application to the FDA, and we plan to make that application in the coming year in 2015. So this is what HD cell will look like. Remember, this is not a currently approved trial. It's a future planned trial. We will be delivering the cells into the brain, and I'll show you with the little arrows where we'll be targeting within the striatum patients who are enrolled in HD cell. And we'll be delivering them through a neurosurgical, a brain surgery procedure, where patients will have a small opening made in the skull in the operating room, and they will have the cells implanted directly into the brain. And there's an example there of a patient undergoing deep brain stimulator surgery for another condition, Parkinson's disease. We're using very similar techniques as are already used for patients with Parkinson's to implant these cells and to then let them go and deliver their payload of BDNF and hopefully create the changes, the positive changes that we're looking for. So I just wanted to transition here before I go back to Dr. Knowlton. We are actually doing a survey, a study, and we would love to have your input if you are a patient or family member or caregiver of somebody with Huntington's disease. We have a survey that with a link there that we'd like to ask you some questions anonymously about your beliefs and your concerns and your hopes for novel treatment in Huntington. So as we've mentioned, this is a stem cell trial using gene therapy that will require brain surgery. So it's a complex trial that we're planning to do, and we'd like to get some feedback from people from the community about how that looks to you and what concerns or questions you might have about that. So I'm going to turn it back over now to Dr. Knowlton to talk a little bit about future directions for our program and for the lab.
Thank you so much. So we're going to continue using cell and gene therapy to deliver genes that shut down the production of or destroy the mutant Huntington RNA in striatal neurons. And that strategy, we hope, will prevent the devastating mutant protein from being produced in the neurons, or at least being produced at high levels. And that is particularly important in the rapidly progressing juvenile HD. And our goal is to have a future clinical trial for juvenile HD when we have enough of the data accumulated in the juvenile HD mouse models with which we're currently working. So we have patented our techniques that we started in 2007. This is mesenchymal stem cells producing inhibitory RNA for disease modification. And these approaches are targeting steps 1, 2, and 3 in this very complicated schematic that you can see. But basically, there's a mutant gene that has the expansion in it. It makes a mutant transcript, that's called the RNA, and then that's translated into protein. And so we have all of these strategies ongoing in our group. We're looking at permanent correction of the DNA in step 1. 2, stop RNA from being produced, and 3, attack and destroy RNA soon after it's made. And other fabulous groups are working on these strategies as well, but we're hoping to use them for juvenile HD. And in all cases, we're hoping that the mutant protein that's killing the cell is not made with any of these strategies. So in our previous work that we've published in 2012, we've shown in the blue line the transfer of siRNA from MSCs directly into neural progenitors can cause significant reduction in the levels of the mutant Huntington protein. And we want to, we're testing this now in the brains of the juvenile HD mice and also in the peripheral, by peripheral delivery, because there are a lot of peripheral problems in juvenile HD. And so we're seeing which one better helps the animals. And again, we have published this strategy, and this goes back to how I had showed you at the beginning with my little video, how these cells can directly infuse into sick or dying cells, directly infuse factors from inside themselves. So these factors don't have to go outside the cell. They put them into the neuron, wrapped up in these little goodie bags or packages called exosomes. And they can also put a little hose, like a little gas hose, directly into the sick cell and pump things into that sick cell. And so that's what well, not a large proportion, but a proportion of our teams are doing, is working on this type of strategy for the future, while the rest of the team is working on the adult Huntington's disease BDNF trial that Dr. Wheelock just mentioned. And so I just wanted to show you again how this happens with the cells, how they talk to each other. They can secrete proteins like BDNF that then get taken up by the other neurons, but they also communicate through touching and direct infusion of factors into damaged cells through tunneling nanotubules and exosomes. And you can see here on the left, there's a red labeled neuron. This is an at-risk neuron because it has a mutant Huntington allele in it. The MSCs are in green. And we'll see a video next of the green MSC infusing the neuron with factors from inside of itself. Without going outside the MSC, it's directly sending those little packages that you can see down into the neuron. So we'll go to the video. Let's see, where's my mouse? There it is. Okay, let's start this. You can see the at-risk neuron on the bottom, and then, oh, sorry. Gosh, we'll go backwards. There we go. Thank you. It's hard to get the mouse to move without starting the next one. Okay, so here's this. You see the at-risk neuron in red, and then the MSCs are in green. Shoot. Okay, one more time. I really want to show this. There we go. Okay, so one neuron, one MSC will come down and touch this neuron and say, I don't know what to do, and run away. And then the MSC on the left will make contact with the neuron and start infusing the factors into it. You can see the green exosomes or packages of siRNA going down into the damaged neuron. And so it's this technology that we've patented and we're hoping to take forward to deliver these genome editing and siRNA factors into the damaged neurons. And this has taken a lot of teamwork. Donors, scientists, physicians, staff members, foundations, community members, patient advocates, the patients in trials. I cannot thank you enough, the families, and the most amazing funding agency ever, CIRM, who funded this as early translational grant. So thank you so much to everyone. It's really a great grassroots effort that has now resulted in a clinical trial, and we hope clinical trials in the future. This is our team, who we meet every Monday morning as the team that all work in the Institute for Regenerative Cures. And I just want to show you how much HD and JHD have taken over my heart, because now this number of us work on HD and JHD. So thank you to everyone. We have an outstanding HD team here at UC Davis, shown on the left, and then our amazing international collaborators shown at the right. And a special thank you to AC patient advocates, patients, and families. You inspire us every day, every minute of every day. And thank you to the California Institute for Regenerative Medicine. That's all. Thank you.
Thank you.
Thanks, Vicki, and thank you, Jan. That was fantastic. I just remind everybody, if you have any questions, go ahead and type them in now, and we'll make sure that they get asked. While we're waiting on some questions, I guess I'll start off with one in regards to the, are there plans to move, to test your mesenchymal stem cell therapy in something to bridge, something larger than a mouse model of HD before going into humans? You know, I know there's a lot of research going on in the HD community to develop mini pigs and sheep and non-human primates. So I'm wondering if there are plans to test the delivery and spread and things like that of these stem cells in a larger mammal.
That is a great question. So we've done, we have a fantastic non-human primate center here. It's world famous. We've done preliminary safety testing of this. It was safe. We do not have an animal model of HD or juvenile HD right now. I'm very excited about the models of the pigs and the sheep, and we'd really like to get them here to our veterinary school, because we have one of the leading veterinary facilities and School of Veterinary Medicine here. And we have not done that yet, but I would love to work with anybody that can help us do that.
And this is Vicki. Also, we definitely intend to test the delivery system in animals, not Huntington model animals, but in larger animals before going forward to the human trial. but that will just be in a small number of animals, and we'll be just looking at the delivery methods and the spread of the cells and the BDNF expression, but we won't be able to look at anything about how it would affect the animal because they don't have Huntington. Yeah.
Mm-hmm. Now, I assume that you would be pursuing a bilateral injections of humans and larger animals?
Well, that's the plan as we've developed it already. I think that that our work, and also I think the work in other diseases, really strongly argues that it would be best to go forward with bilateral injections of the cells and not just unilateral injections. But I know that we have to engage the FDA to discuss that because it is possible that they might ask us to do unilateral injections. I think, again, we like to do bilateral, but it will depend on what our rationale is and how well we present that to the FDA.
Yeah, the bilateral really strongly increases the potential benefit to risk ratio.
Absolutely, yeah.
So we're getting a number of questions coming in from the audience. The first is, what are the risks? What risks are there for these phase 1 patients?
Well, several things. First of all, we think that these cells are very safe cells. We think that the gene therapy is very safe so far, but we still have studies underway to really demonstrate that safety in the animal models, in the Huntington's transgenic mice. So we have work going forward to do that. I think one of the most obvious concerns would be this is a brain surgical procedure, so there would be a potential risk of having brain surgery. The risk of brain surgery could include a little bleeding in or around the brain, which could cause a stroke. We think that the risk of that is very low. It's certainly very low in doing deep brain stimulator implantation. But that's another reason that we're going earlier in Huntington's disease, because it seems that the risk of going later in the disease of doing brain surgery could be potentially concerning or excessive with respect to bleeding. We think infection is very unlikely. We think that an immune response to the gene-modified cells is very unlikely because of the innate properties of the stem cells, but it's possible that there could be an immune response within the brain. So that's something that we are doing tests to look for, and we will be monitoring extremely closely in the patients who have the therapy. Those are just a few of the things to think about. Again, I want to also emphasize this is a safety trial, so we'll be looking specifically for any signs of complications or side effects. We'll be seeing people weekly and biweekly and very frequently to monitor them as closely as we can going forward. The last thing I'll say is there may be some additional safety concerns that we don't know about yet because there's still some studies that are ongoing in the mice. So that's what I can share with you right now.
Another quick question is, would you be willing to, or making the results of your survey on HD treatment, your HD treatment survey, available outside of CIRM? Would you be willing to share?
Yes, absolutely. In fact, we have a flyer about the study, and it says on the flyer, we will be sharing the results of this survey with the Huntington's community. One of the things that's so important to Jan and to me is to make sure that we're communicating and engaging the community as often as we can about this. So that is absolutely the intention. I think it will be very helpful to us in designing HD Cell to know what things from the community might be of concern. But I also think it will be very helpful just to show to regulatory agencies, just as you are with the FDA and with CIRM, about what people's beliefs and hopes and concerns would be about potential participation in a study like this.
Great, thank you. So we have a couple different questions regarding the pre-cell clinical trial, mostly around recruitment and eligibility. Is pre-cell willing to accept out-of-state subjects if they're able to arrange their own travel, and are there costs to the subject for pre-cell assessments?
So the answer is we are open to people from out of state. You don't have to live in California to participate in pre-cell, but unfortunately we are not allowed to support travel for people who are in pre-cell, and that's been frankly a big limiting factor for a lot of people. So because in pre-cell we have visits every 6 months going forward, but we're also asking people to think about if we do succeed in getting the IND to go forward to HD Cell, that will require very, very frequent and close monitoring. That would preclude maybe going back and forth, every week. So we're kind of trying to give people the big picture about out-of-state participation, but I do want to say we do have people from out-of-state who have volunteered for pre-cell. We are hearing from people about that, and we're doing our best. Terry Temkin, who's our clinical project manager, is very, very involved with people, helping them to arrive at a decision that would be good for them. I wish that we could help support travel, and I'm sorry that we can't.
And I wanted to add that aside from the travel, there, of course, is no cost to be in this trial. Places that charge you to be in a clinical trial, there's a big problem with stem cell tourism. And so just always beware. And if you're worried about something like a clinical trial that's asking you for a large amount of money to be in that trial, just send me an email and ask me about it, and I'll give you maybe some insight as to whether that's a legitimate place or not. Because there are a lot of places outside of the United States that prey upon people who are desperate for stem cell trials, and it's a big problem.
Right.
Yeah, thanks, Jan. So again, no charge for any of the scans, any of the tests. All of those things are provided by the study. Yes.
Great. Another question. Other than the adverse events associated with the administration of the MSN, or sorry, not MSN, but the mesenchymal stem cells, what side effects are expected given the data we know now?
Well, at this point, we're not expecting any particular side effects from the cell administration or from the BDNF expression, but obviously that will be a question going forward. That will be something that we're going to be looking at. At this point, we're not really expecting any particular side effects to happen, but let me be very clear. We're going to be going into a very early phase novel treatment, and it will be our job to anticipate, potential side effects and to watch for them very, very carefully, and we will be doing that.
And also, there have been a large number of MSC clinical trials for other indications because these are good helper cells and paramedics, and we have not seen severe adverse events in any of those trials, and there have been a couple where they have been delivered into the brain. So there is a record of safety with these particular cells that, again, we will monitor very, very closely.
Oh, that's great. That was one of my questions. I'm not familiar with the field that much to know if there have actually been proof of concept studies of mesenchymal stem cells actually going into patients' brains.
Yeah, there are two studies in the brain, so not for Huntington's, for other indications, stroke.
Another great question is there any potential for injecting these cells via like a lumbar puncture or spinal tap, similar to what I guess they're going to, ISIS and Roche are planning to do for their ASO studies, to kind of bathe the brain in supporting cells?
We think that, in fact, that's a very good question, and believe me, if this could be a pill, I'd even be happier. If we could just give people an MSC pill, an oral therapy would be ideal. But we believe, based on how these cells work, that we have to deliver them directly into the brain. We think that the blood-brain barrier, that even within the spinal fluid itself, will only allow penetration to some of those areas that are close to the surfaces. You have to have inflammation in the brain for things to pass easily from the CSF. In other words, something as large as an MSC. And so we feel that we have the best chance of helping the patients by delivering the cells directly into the brain. And again, if we could do it in a simpler platform, we wish we could. We don't see an avenue of doing that right now.
Okay. Another question is, do you expect or suspect that the bone marrow or stem cells that you'll be using be required from close family members to be effective, or can it be just any unaffected control person?
Yeah, that is a great question, and we've had family members offer to donate because they're so invested in this research, but there are a lot of qualifications for the donors, and there's actually an FDA guidance for what these donors would be. They don't have to be related. That's a pretty incredible thing about these cells. They shelter themselves from the immune system and won't undergo immune attack, and so we can use them from a selected non-related donor. And that's what's happened in the other trials that I had mentioned earlier. It's called a third-party donor because it's completely unrelated.
If another question is, if all goes well and they're safe and seem to be moving the needle in the right direction from an efficacy standpoint. Is this something that patients would require over the course of a lifetime, multiple surgeries, or is this a one-time deal?
At this point, with our phase 1 trial, we're going to be just doing a single surgery, and that's actually another very good question. We hope that delivering BDNF at a point in time, as we're doing in this trial, will help to rescue some of the sick and dying medium spiny neurons and other neurons that are affected. We hope that we will recruit some new some neurogenesis, some new cells to help restore this area. But we will have to be studying in the patients exactly how long the benefit would last. And I would think, to answer your question, I wouldn't think we would be doing repeated surgeries. I think we might be looking at another delivery system where we might be able to deliver the cells through a catheter or something that would be implanted, and that would be done periodically. Again, it remains to be seen how often that would have to be done.
That's great. There was a recent paper, I think it was earlier this year, that looked at brains of patients that had received embryonic stem cells. And I think that some of the take-home was that they found that those unaffected stem cells actually got sick, and they found Huntington aggregates in the unaffected cells. Is there any evidence in your preclinical models, in the 2 different mouse lines that you're using, that your mesenchymal stem cells are getting Huntington?
Yeah, it's a great question, and thanks for whoever asked that question. There certainly could be two-way flow of direction. So I showed you that the cells will basically open a tube into the other cell, and things don't just flow from the MSC into the tick cell, but can kind of retrograde flow back into the MSC. And so it is possible. The MSCs are around in large quantities for just a few months, and we hope that they do their job as well as they can and deliver what they need to deliver during that time period. And that's how we're focusing our trial before they could get sick.
Right. So these cells are the delivery mechanisms, but they're not really intended to grow to themselves to last for years and years like an embryonic or an induced pluripotent cell would. So I think that helps to answer the question, too. We think that they'll be able to effectively do their job before they might be affected by the mutant Huntington protein.
Okay. So, what's the advantage of this route? Maybe you can just walk us through the advantage of having cells, mesenchymal stem cells, act as a delivery device for BDNF, dystrophic factor, versus just direct delivery of say, an adeno-associated virus that overexpresses BDNF. What's the advantage of going the mesenchymal stem cell way versus just direct gene delivery?
Sure. We're also big fans of the AAV systems, and I hope that this all works. I hope anything works for Huntington's. The reason that we focus on MSCs is that we think it's safer. So the AAV goes directly into the neuron, and then if anything would go wrong, that neuron's at risk. With our gene delivery system, the exogenous gene, and especially for our siRNA that we're delivering in the future, the exogenous gene is just safely integrated inside that mesenchymal stem cell. And so if the cells need to be killed off or anything might go wrong, it's only the cell that we put in. It's not further damaging the brain. But I'm not saying that AAV would be damaging. It's just a different strategy to get our molecules into the brain. And the MSCs travel, as you saw, quite nicely throughout the striatum, rather than just going into the area of injection.
Again, the MSCs have some innate neurorestorative properties also. So by reducing inflammation and by helping to downregulate programmed cell death and a number of other mechanisms, actually, it can really, we think that we see an additional advantage of using the cells for that reason also.
Great, thank you. So you mentioned biomarkers. There's one last question kind of related to that, and I think I saw a slide in there. You were talking about the different things that patients undergo in the pre-cell program over the 18 months, the different visits. You mentioned something about CSF collection and biomarkers. Can you comment on what kind of particular biomarkers you're taking a look at as potential readouts for efficacy?
Well, the most important biomarker is going to be BDNF levels, clearly. But actually, one of our really valued collaborators is Steve Hirsch, and in the Hirsch lab they're doing a number of studies, of exploratory studies about biomarkers that we hope to link up with pre-cell, with the clinical measures, the volumetric measures, with looking at both blood and CSF. So there could be a chance to look at exosomes. There could be a number of other things that we could be collecting within the CSF. So we're looking to the leadership from the Hirsch lab to help develop some exploratory measures.
Great, thank you. One last question, and it's kind of a follow-up to the survey question that someone else asked, was where will the survey results be posted when available?
Well, good question. So we go to meetings. We're going to the Huntington's Disease Clinical Research Symposium in Minneapolis in about a month, and we'll be presenting information about our study there. So it could be there, it could be at our local chapter convention. If you at HDSA were interested in having us come to national convention, we'd be happy to share it there. But we have a website in our Huntington's Disease Clinic that we could post information. So we will get it out there. I will tell you, when you look at the survey, it's going to take us quite a while to analyze it because there's a lot of open-ended questions. So we are assembling a team of people who can go through that, and we want to make sure that we have a really valid analysis. But again, there are a number of venues where we could share that information.
Yeah, and I can say we're happy at HDSA to share any of that information as the data are final and you have a nice conclusion. We're happy to share and promote and make sure the community is aware that the results are out there and post it on our website or wherever if that helps. So we're out of time and right on time. So I just want to thank you guys again for taking valuable time out of your busy schedules to present on this very important topic. I'm sure that I speak on behalf of the entire community. We thank you for all your hard work and your team's work, and thank you again for filling us in on this very exciting program. Thank you very much, and look forward to talking to you soon.
Great. Thank you. Thank you. Bye-bye.
Bye.